I'll bring some concepts and then we will run a script together. We have here in the main page the tutorial. Here there's more text, a lot of information. And here is just the code that was extracted from here, which is the code that we will run today. Okay, so today we talk about exploratory data analysis, and that involves descriptive statistics, exploratory graphics, and variable relationships. The idea of exploratory data analysis was proposed by Tukey, and this is a 77 book, probably, something like that. It was published in the 70s, and it was the first time that someone organized information on why we should do that and general suggestions of how we should do that. There are more recent references, but I like to bring this because this is the base for what we do today. We have that in our we have all the references listed in the main page, and that there's a Zur book of exploratory data analysis that is more recent. But the main idea of exploratory data analysis is to know your data really well. So it's always a good idea to spend a lot of time understanding your data whenever you can. And the goals of the exploratory data analysis is to control data quality, suggest hypotheses for observed patterns, maybe something that we haven't think before yet, support the choice of statistical procedures for hypothesis testing, and assess whether the data meet the assumptions of the chosen statistical procedures, and indicate new studies and hypotheses. Why am I saying all of this? Because exploratory, when we are analyzing data, the first thing is that we need to have an hypothesis. And that means that exploratory data analysis is not data torture. So we're not, after we collect the data, we're not trying to extract something new. We collect data because we have a question, we have a hypothesis, we have previously designed how to answer that question in terms of statistical methods. So we should think about statistical methods first and collect the data in a way that we can analyze the way we planned to. So it is assumed that the researcher has a hypothesis prior to collecting data. Okay. Sometimes we work with secondary data, and then the data was collected, and then the hypothesis comes second, but the idea remains the same. Okay. So imagine, like, when we are working with secondary data, we formulate a hypothesis, assuming that the data was collected in a particular way. Okay. And the thing here is that there's no recipe. There are like tons of guides of how to do, but it really depends on the nature of the data that we have. And usually it takes a lot of time, and most of time that we are analyzing data, we spend in the exploratory data analysis. It can be started during data collection, and it's advisable to do that because we can decide in the middle of the process if we need more samples or more treatments or something like that, if we need to change something in the middle or in the beginning of the experiment or the sampling. And visual techniques are widely used, so Understanding a data means that we need to know what is on the spreadsheet, but also visualizing it makes a huge difference, and I will show you that today. I'll show you now, actually. So, Francis Anscombe was a mathematician, and he created a data set to show the importance of visualizing data. And he created 4 data sets with the same descriptive statistics, but they are very, very different graphically. Okay? We can load, the data exists on R. It's on R base. And when a data set exists on R, we can load the data set to our environment using the function data. So we are using data, Anscombe, and then Anscombe data is on our workspace. If we check the variance of this data, we have x1, 2, 3, and 4, y1, 2, 3, and 4, and the variance is basically the same between the x, 4 different x's, the 4 different y's. Here I wanted to show you the mean, but I didn't do that. We can see that in another way. But let's take a look into the data. So the first 3 x's are very similar, but the y's are always different. And if we take a look into this, we could say like they are not equal, right? But if we try and see other metrics, the correlation between x and y for x1, y1, x2, I'm asking the correlation between x1 and y1, x2 and y2, x3 and y3, x4 and y4. They are all basically the same. They differ after the 4th decimal case. They are basically equal. If we apply here, I'm applying a linear model to x1, y1. I'm doing the same thing for the other x's and y's. I'm creating a linear model, and then I'm using the coef function to extract the coefficients of the model, the a and the intercept and slope of my model. And again, they are all basically equal, intercept -1, the slope 1.33. But if we take a look graphically to this data, they are completely different. So it's super important to understand the mean, the deviation, and descriptive statistics of the data, but we cannot do just statistics. We have to actually look into the data because there's a lot going on there, and we wouldn't see that if we weren't plotting this data here. So some guiding questions for exploratory data analysis is where the data is centered, how the data is distributed, are the data symmetrical, asymmetrical, bimodal. We'll talk more about statistical distributions next week before the linear model, the statistics the modeling module. But right now we will see data distributions and compare with a normal distribution today. This should be part of the exploratory data analysis, but it's also a preliminary part for any statistical modeling that we will do. We should always check for outliers, if the variables follow a normal distribution, if we need that way, or what is the distribution of the variable. We should check for relationships between variables, and if those relationships are linear. Do variables need to be transformed? And this is a super important question. Was the sampling effort the same for each observation or variable? Sometimes it is not, but if it is not, we can do, there are statistical tools to deal with that. But sometimes you need to assure that this is happening, and the sampling effort was actually enough, actually, for what we want. That is like the gold question in statistics. What is, okay. Use the microphone.
Okay. I just have a question. When you say sampling effort, what does it mean exactly?
Okay. When you have a research question, and you need to answer that. Sometimes we do experiments. Sometimes we do sampling. And for both of them, we call sampling effort, but it is the same idea for experiments. It's basically the number of unique observations. Your sample unit, every time you have experiment, you make experiment, you have 2 treatments, 30 sample units for each treatment. And your sampling effort was 30 for each treatment. Was that enough to understand what you want? So it's basically how many observations you have. And if you have, and it will really depend, like if it is a simple question and you are doing, I don't know, you are trying to understand if a plant species can survive to flooding. So you get like, you get 30 plants and 30 not, but like 10 plants, and you don't add water, and on the other 10 you add just a little bit of water, and then you increase that, and you have like 4 treatments with different amount of water that you are pouring into your plants. And your sample unit there is each vase for each treatment. So when we are evaluating if the sampling effort was enough, it doesn't matter if I have 1,000 of plants. I need to distribute that into my different treatments. So the sampling effort will be related to what is your effort per level that you have, okay, in an experiment, or per plot if you're doing a sampling. But it's how much the number of observations per experiment or per sample.
You wouldn't want to have like 2 plants with no
Yes.
water, and then like 20 for the ones.
That's not a good idea for experiment. Sometimes things happen, like people work with germination and plants die during the process, and you end up in the end of your experiment with different numbers, different sample units for each treatment. And then you have to do that with statistically. There are ways of dealing with that, but in general, it's advisable to have like equivalent sampling effort.
The 0.6 that it was previously in the presentation, it could be thought like batch effect, like how can we correct batch effect? That could be the 6th point, like sampling effort, the same of each observation. It could be like thought like batch effect.
For instance, you can deal with that with mixed models, and you would have a random factor describing each treatment, and that way, like, you can assume that you don't have the same number for each treatment. That's one way of dealing with that. If we are doing like parametric statistics, you can sample, like you have like 1,000 in one treatment and 500 in the other one. You can sample 500, random sample 500 from the 100, and then it will diminish your sampling effort, but sometimes it's the only way of dealing with that. So you can either sample or like deal with that statistically. But I mean, we need a hypothesis, and the sampling design class is not today. So we need to assume that we have a hypothesis and that we delineated our study in the best way that we can. So there's, I don't know who, a famous statistician that said that every time that people knock it on his door, asking them, oh, I have like my experiment went wrong, and or even if it didn't went wrong, but the sampling or the experimental design was terrible, and then after they do not think about consulting a statistician to think about the sampling design or the experimental design, and just knock on a statistician door for the analysis only, and the statistician can only say from what the experiment, why the experiment died, or why the experiment wasn't enough to answer that question. So sampling, designing an experiment and a study is super important, and we need to think about design first, not at the time that we are analyzing the data.
I mean, if I'm using data that I didn't collect it, and one group have a large number of samples, and the other group have not a large number, a small number, but I want to compare these two groups. What's the best way to do? Reduce the number of the other group, number of samples, or just do in this way?
It really depends on to sample and reduce your sample. You would need like a reasonable amount of observations. If you do not have that, I would say that the way of doing that would be with a mixed model. So you can deal with the unbalanced sampling design or experimental design. But it will depend on the data, really. But you can either sample or use another statistical technique to do that. If you like, there are premises from like most of parametric statistical tests, so you cannot do a t-test if you have unbalanced samples. But you can do like a mixed model. Okay, so a bit about descriptive statistics. And besides like metrics, I'll talk about other questions. So first thing, like we talked a bit another time about missing values. So it's really important to check the missing values in your data if you have. And check if they are really missing, if sometimes people make a lot of confusion with NAs and zeros. And sometimes like NA is a zero, or it is just an information that is not available, actually not available. It's important to check that. Check for zeros, because the opposite can occur too. So sometimes the zero is information that we don't have. And sometimes I will show you the example with the census data that we are working with. And when we deal with abundance data, for instance, we have a lot of zeros. And that's correct. But there are other situations in which, like, if we have a lot of zeros, something went wrong. So we need to make sure that NAs are NAs. Zeros are zeros, and they are real. where the data is centered, how they are spread, are they symmetrical, skewed, bimodal, do we have outliers, and what is distribution of the variable. We will use, to answer those questions, we will use descriptive statistics, the mean, the arithmetic mean. In R, we use the mean function, the median, we use the median, the mode is the most frequent value. We can use the function table to count how many observations we have, and then sort them from the most frequent to the least frequent and get the first one. This is the mode. The standard deviation and the quantiles. The quantiles are used, we will see further in this class, how we can cut a distribution in parts. Sometimes those parts are equal. If we are dividing into 4 parts, we have a quartile. This is a specific quantile division, but we can do quantile with other distributions. And we will do that in the script. And this part that is super important, exploratory graphics. I will read here the Poisson data that we generated yesterday and the environmental table. I'm getting only, I want a table only with the environmental variables. I'm removing the site column because I want to perform a lot of things with the variables only and not with the column site. So this was what I was talking about. So here I have a box plot of the abundance data. I cannot see the box plot. This is not wrong. This is because the distribution is definitely not normal, okay? A box plot is super useful to get a first look into the shape of the distribution, a box plot and a histogram as well. So if we check the data abundance, we will see that the median is 0, the third quantile is also 0, and the mean is 0.1. The maximum value here is 6, so it's like we do not have like very abundant species, but we do have a lot of zeros. We can count how many zeros we have, so I'm doing a logical test here to check how many values in abundance equals 0. In R, a logical vector, true and false. True equals 1 and false 0. So if I sum this, I will get how many true values I have here. So I have 4,000 zeros, almost 5,000. And I can check the proportion. Oh, this is cut. But this is basically 88% of zeros in my data. And how do we interpret a box plot? This is a box plot. Now we can actually see the box plot because this was built from a normal distribution. In R, by default, the strip here is the median. It will depend on the software that you are using. Sometimes we see box plot where this here is the mean, but in R it is the median. And we have here the first quantile is the bottom of the box. The third quantile is the top of the box. The length of the box here is the interquartile range. And the extremes are the last point times 1.5. Minus 1.5 and plus 1.5. This is a general rule for establishing what outliers are. So everything that goes beyond this upper limit or this lower limit is an outlier. So those dots that we are seeing here are outliers. And we can extract them when we use a box plot. So we can box plot a variable and plot them. And if we do box plot and plot equals false, we will get the information of the distribution, and we can actually find the outlier, where it is in our data. We will do that. Another good way of visualizing data is using a histogram. This is much better. I like a lot histograms because it works well for any distribution, not only a normal distribution as a box plot. And what we have here, we have different bar widths. We will see how this change. But this is basically how many observations I have between 0 and 0.5, and 0.5 and 1, and so on. And here, basically, most of my observation, actually much more than, it's almost 90% of my observations are 0, so this bar is really huge. And here I have the frequency, how many observations follow into this interval, okay? This is a histogram. And this is continuous. This is not a bar plot. The difference between a histogram and a bar plot is that the histogram is continuous, and the bar plot is for categorical variables, okay? Here, we have two types of histogram, the frequency histogram and the density histogram. They will look the same. The difference here is only the y-axis. So here I have the frequency, and here I have the density. For doing that in R, we will only do hist is the function that will build a histogram. So I'm building a histogram with the column abundance from my all data object. If I set probability equals true, it will plot a density histogram here. The difference is only the nature of this data. This is already the empirical distribution of the data. And when we want to check what distribution best fits my data, I will always use a density histogram or a density plot, because this is the description of the empirical statistical distribution.
This density distribution should be not delimited by 1? Or is other thing like 1 as 100%? Or is not this?
It should not, because we are seeing that.
Yeah.
Sorry. How can I explain that? I will show you that next. Okay.
Well, I think that what it had to be 1 is the sum of all the areas. So
yes, exactly.
That's why it's not necessary, because in there, if you see the box, well, the higher rectangle, its base is 0.5 or something like that. And if you calculate the area of this rectangle,
yes,
it's less than 1. And then you have
yes,
to add all
yes,
of them.
Yes. You have, thank you for that, because if we have the actual curve, what describes the empirical distribution is the curve, and the area below the curve is 1, always. This is just the frequency. Thank you. The classes, we have different classes. We can control the width of this bar here. So we do that by using the function, the argument breaks in the histogram function. So all data, I'm still doing my histogram with my column abundance here. And I'm setting my breaks from 0 to the maximum value here. And I want to divide into 3, so in intervals. So I would have here only 2 bars. If I'm dividing into 5, I would have 4 bars here, and here is the default. So it does like the ideal distribution for the most recommended way for observing your data, the default here. But we can control that, okay? We'll talk about this command probably tomorrow. And here I'm setting the default in R is always to plot on one device. I want to plot here 3 plots on the same device. So I'm defining that I want to create an empty box with 1 row and 3 columns. And I'm adding each histogram to one of these columns. And then if I do this, my entire R section would be with 3 columns. And I do not want that. So every time I do this, I will do this MF row 1, 1 to get back to the default. Okay.
Yeah. The length, can you be back? Yes, the length is 3, right? But I just count 2 bars. I don't know, the length.
Yes, the length is the limits. So we have 3 numbers. We will have 2 intervals.
Oh, okay.
Okay. So I'm basically defining what are the marks here. So 0, 3, and 6. If I do this, here is 0, whatever, 1.5, yes. So the empirical probability density curves will represent the function that describes the probability of finding a certain value. So when we do this, we have a histogram, a density histogram, and this would be the equivalent of this visualization. And here, the area here would sum to 1. And we can overlap a density curve to the histogram to visually check if the distribution matches, fits the data. So we already saw that our abundance data are not normal. So one possible distribution for that would be Poisson distribution. This is count data, a discrete distribution for count data. The parameters of the distribution, Poisson distribution has 1 parameter, lambda, which is the mean. I'm calculating here the maximum because I would need that for plotting, and the mean. So I have my maximum of abundance and my lambda here. And the way I create a density distribution is in R is using the Poisson function. We have function for the most common in base for the most common distribution. It will always be D and Poisson. So this stands for density and Poisson for the Poisson distribution. We have a D norm for the normal distribution, for instance. And we will plot from 0, a discrete distribution from 0 to the maximum of my data with the lambda parameter that I observed in my distribution. So this is my observed parameter. And I'm defining here, this is something that I did. I defined a vector of colors, and I'm in the beginning of my script, and I'm grabbing the 5th element of that, and this is not red. Like I wanted a specific red that I defined. That's why I did that. And I'm plotting, so here I'm plotting a histogram. We have plot functions there that are secondary functions. Points is one of them, lines is one of them as well. And I plotted a histogram, and I can add points and lines to that just by calling a secondary plot function after my main plot function. So if I start my graphic with points, it won't work because this is a secondary function. It will only work after I have a plot device. So I have my histogram. I want to add points, so I'm adding these points here. And I want to add lines, so it's better to see. So I'm adding also lines with the same command inside. This is exactly the same that I have in points here, but I'm building lines. I can easily see that it is far from being a Poisson distribution because it has a lot of zeros. So there are zero inflated distributions, and abundance data usually follow those distributions. But this is for us, this is one way of checking if our particular distribution fits our data. There are specific packages to do that, but in our base, we have to do that by hand. And I like doing that by hand because we can understand what the parameters of the distribution are. And we have like the most distribution that we are most familiar with is the Gaussian or normal. It has two parameters, the standard deviation, and the mean. So here I have three distributions with the same mean, but standard deviations that are really different. So this is another tip for us to never stick with the mean. We always need a deviation metric to really understand how our data is distributed, because it can have the exact same mean but completely different distributions. And this is just an example of why sampling is important. So I have here, I'm sampling here, I'm randomly generating data from a normal distribution from, I don't know, a little bit less than 5 to a little bit more than 10. And it has a particular SD and mean. It has the exact same mean and standard deviation, but this is not 200. This is also not correct. But here I have a 200 sample. Here I have for sure less than that, and here I have much less than that. So if I compare those empirical distributions with the empirical distribution that I would have if my data followed a normal distribution, when I have like a big sample, this is like, okay, this is normal. This is very acceptable for a normal distribution too, but this I would start doubting if this is a normal distribution. So this, we have like a continuous data that should be normal. This would indicate us that we are not there yet to describe our sample. So another thing that we want to explore is relationship between variables. So I read my environmental data in R. So I want to see what is the relation between clay and silt. Those things are correlated. We know that, but I want to see how they are correlated. So a scatter plot is a really good way to see the linear relationship between two continuous variables. We can calculate the correlation between those variables. So here I'm applying the function core to my entire table. So we do not need the apply here because the core function is a smart function that can deal with a vector or a table. I have here the correlation for my environmental table, and when we apply the function correlation to a data table, what we have here is a matrix. So The diagonal of the matrix is 1 because the correlation between clay and clay is 1, of course. And they are symmetric, right? So clay and silt is 0.62, silt and clay 0.62 as well, okay? We can either check the upper or the lower diagonal here, but this is how we can get the values, the correlation between all variables that we have. The idea of looking into correlations is to see how close to a linear relationship two variables are. So this is a correlation that equals 1. Those correlations are very far from a linear relationship and they are 0. This is -1 because it goes into the opposite direction as this one. And as we go further from the linear line here, our correlations are slower, okay? So this is how we can interpret the correlation between variables. A thumb rule is that any correlation higher than 0.7 is a high correlation, and we should be careful with that, but it really depends on the nature of your data and on the area that you are working with. Sometimes correlation that is 0.6 is already something that you should be careful with. In our base, we can visualize those correlations with the function pairs. So this is like doing a scatter plot for my entire matrix. Again, this is the same structure as in my correlation matrix. So I have a diagonal here, and this is good because I have the name of the variables, and I can check the correlation between clay and silt. Silt and clay would be the mirror of this, the same thing as this. And we can check like any relationships that we want, like silt and magnesium. It is not really correlated. This is a good way to get an overview of correlation between several variables. There's a package called GGLE, and it makes something similar with this, but with even more information, because I do not need like all this information here because this upper diagonal is the same as this one. So I can use that space to bring more information here.
In the previous slide, we can see that like the correlation between clay and salt, we have almost a line, but also we have kind of outliers point.
Yes.
So
yes.
We can define as an argument outliers true and the remove?
No, this is the outlier. We use the term outlier for one data. So for a within one data, one variable, we can check if there's an outlier because we have one variable and it should have a particular distribution, and we would find outliers by comparing the value with the extremes of the distribution. So outlier is something that we find for one distribution only and one group of data only. But this is saying to us that the variance across the variation of this relationship is not equal as we go, because if it were, we would have a pattern like this. So this is showing to us that for some values, this deviates from the expected correlation here. So it's a deviation from the expected perfect correlation. It's not an outlier, but it's a deviation, and it has a particular pattern. So this is something that is like worth noting, because this deviation is biased to this side, to these particular values, okay, not for the upper case. Here, I can see the empirical distribution of each variable and the correlation, the plot of the correlation and the correlation actual value here. It is even showing us, like we have here these signs indicating if it is significant or not. This is the least important thing in this visualization. I would stick to the graphics for now, and if you need to perform a test to check if the correlation is significant, you can do that, but this is not the most important thing here.
I heard that making many correlation tests and statistical tests could be leading you to a statistical error, like something like this. But I don't know what's the limit, like how many comparisons and correlations can I do until I be doing something wrong, something not good for science, you know?
Okay, so I'll address that question next week when we are talking about statistical modeling. But the important thing is that there's a limit. We have different types of doing statistical inference. We have the classic way of doing statistics. We have likelihood methods. We have Bayesian methods. So if we are thinking about parametric tests, we have to assume a lot of things before doing that. So your variables should be normal. There are a lot of assumptions here. We cannot have an unbalanced sampling to perform a correlation, for instance. And there's a limit of significance tests that we can do. And you need to calculate your p-value differently if you're doing recursively correlations. So there's a limit, of course, and you should perform like one test to answer one question, basically. Like we do those things to explore data. Maybe I want to do an analysis and I want to include all those variables. This is a good way of thinking, like if I'm including clay and silt, I'm adding two variables that have the same pattern, and this would be almost like a pseudo sampling. I would be like repeatedly doing the same test, and I don't want to do that. So there are ways of dealing, like if I want to like work with all of those variables, we can do a PCA to reduce the dimension of all those variables into one variable that is definitely not correlated between each other. So the amount of variables that you will deal will depend on your question, will depend on how they correlate to each other. And if you need all of them, you have to treat them somehow to do that. And you should not be doing like tons of tests for the same question and I have like 5 different p-values for the same hypothesis. No, you have one hypothesis, you have like one p-value if you're using that kind of inference. And there are ways to correct the p-value if you're doing like different tests for the same hypothesis. Okay.
And how can I know the better value to include or exclude one variable in using the metrics? It depends of the test I am using before.
To exclude a variable that is like that has a high correlation. That's your question. Okay. We use that cut of 0.7 like as a cut, but it will really depend. There are also ways of, like, sometimes, like, if those variables are super correlated. They're sand and clay are super correlated here, okay? Minus 0.7 here. It will depend, like, I will need to remove one of them. I would stick with sand or clay. I don't know, it depends on what I'm working with. Is sand more important to explain a particular biological pattern than clay? I don't know, sometimes we have several variables that are high correlated, and there are tests that we can do how we can remove the fewer variables to still have and remove all the high correlations. So there are ways to optimize that, which variable I would remove, but those tests do not see the biological meaning of those things. So you can do a test to actually tell you remove this, this, and this, if you want to stick with those variables. But it will depend on your question in the end. Sometimes, like, if you remove just one variable, maybe that variable is the most important for you. It's better to stick with only that variable and remove all the other ones. But it will depend, for sure. And there are statistical tools to help us decide, but decision is on us, always. And what are the paths for data analysis is always our hypothesis. So after the hypothesis, what we can do, we really need to understand data well, and that's when exploratory data analysis enters. If our response variable is normal, we can do a linear model and other parametric analysis. If our response variable here has another distribution, we should use a non-parametric analysis or a generalized linear model. If we have hierarchical predictor variables, we should be using a mixed model, either a linear or a generalized linear model. If we have pseudo-replication in space or time or unbalanced designs, we would use a mixed model as well to deal with that. We will cover linear models and a little bit of generalized linear models next week, but those are like suggestions. We always should have, like, we think about our questions, we design our method to answer a question. We would think about the analysis first. We should have like a clue of like what we will do with the data, what our packages we will use to analyze that data. And after we collect, some things can change, and they do change, but it shouldn't change drastically, because we should collect the data thinking about our question and thinking about what is the method that we will use to answer that.
Yeah, I just want to know what is the 4 and 5. I don't. What is that like?
Generalized linear mixed model.
No. The hierarchical predictor variables and pseudo replication is per.
Hierarchical predictor variables. Okay. When we are doing, I will draw that because it's. Like, I will give you an example of how my sampling design for my PhD was. I was studying biological communities in 3 different mountains. So I had 3 different mountains here. And I have a sampling point. I was trying to understand how altitude would affect community structure. And I have here 1, 2, 3, 4, 5. Okay, one more here. I have here 3 different mountains. I have one 5 plots at each mountain. Total, I have 15 plots. But the thing here is that like those plots, they are not independent from each other. A sample unit is a unit that is completely independent. So this is not independent. So I do not have 15 samples. I have basically like 3. And I have subsamples here. This is a typical hierarchical design. This is super common in ecology. When we have experiments, we can have things like that too, a similar structure. So this is a hierarchical design, okay? So when you do not have, this would be, if I treat this as a 15 unique observations, I would be doing pseudo-replication on space, okay? So usually when we have hierarchical design, we have groups of sample units that share something, and because of that, they are not independent. Okay, and when we have pseudo-replication as well. So what we will do today now is that we have not that much of time, but I will go through the script with you. We'll do the same thing that we did yesterday. Click here, we will open the script. I will copy the script. I will open my R project. I already did that. I will paste the scripts here. I will save this as in my R folder under the name of for EDA. Okay. I don't have a particular package here. I will install. Hopefully this is. So we'll begin with our header. This script, it's not like really well commented because all the comments are here in the tutorial. So there are a lot more explanation here, and if we don't have time to go through all of this, I will skip some parts. I want to show you how we can find the outliers, but there are a lot of good explanations here, and we can go and check at least for outliers and compare with a normal distribution. So I will load here a data set that also is on R. This is a data set called IRIS because this is information on IRIS species, 3 different IRIS species, and we have, this is a flower, okay? We have different morphometric measures here. So we have sepal and petal, those are different parts of the flower. And we have the length and the width of both of them for each species, okay? This is the structure of this data. I can check the heading of this data. I can do a summary of this data. I can see the mean values, the minimum, maximum. The species here, we have 50 observations for each species, setosa, iris setosa, iris versicolor, iris virginica. I will show you here 2 functions that are useful for exploring data as well. First one is table. It basically counts how many observations we have of a particular vector, or like it can be a numerical vector as well. But this is the same as what we were seeing here. Like we have 50 observations of each species, but we are doing that with the table function. And I have here two functions that I want you to show. We already learned using the apply function. We used lapply as well. Here we will use tapply. This is useful when we want to apply a function, but we want to do that not to the entire data, but conditional to a particular factor. So what does this mean? I want to extract the mean value of the sepal length per species. So if I do only mean of iris sepal length, this is the mean value including all species. If I do this with this tapply, I would have the mean value for each species. Okay? So how we do that? The first argument is the vector that we want to calculate something. The index should be a list. That's why this is a list here. This is not a list. This is a column from a table. So I have to set this as a list. I could add more to this list. So it's not restricted to one. So my variable, the condition that I want to group the variable, and the function that I want to apply. Okay. The function aggregate does the same operation. The arguments are a little bit similar. So in tapply, the first argument is caps x, in the aggregate is small caps x. The argument for where we specify the grouping factor is, it should be still a list, but it's called by and not index, and fun is the same here. If I do this, I have the exact same information. The main difference here is the output, okay? Is how we specify the arguments and the output, basically. Which one should I use? It will depend how you want your data. I really like aggregate function, is one of my favorites. And we can do that using a different notation. So this is the same function, still the same operation, but now we can actually name the columns here. The notation for that is the column name, and here, this is the same notation that we would use for a linear model or for a graphic. We could use that notation. So it is basically in a graphic, the y-axis, tilde, species. And this is how we do that in aggregate. And we have to specify from which data. So those are columns of some data. We have to specify which data and which function we want to use. So it's basically the same output that we had here, but now we have better column names. We should always check for NAs and zeros in the beginning of our exploratory data analysis. I will here apply to my data iris to the columns, because I'm setting here this argument to 2. This is doing applying a function to columns, and I want to be. I'm using a function that I'm building. I'm not defining. Oh, this is not good. I'm not defining a function in my environment. I'm defining the function inside the apply. This is something that we can do. This is useful when we do not want to create another object in our environment. I will just use this here, and it's a super short function that can fit into one line. What I'm doing here is that I'm summing. I will do that for one column, for instance. So is an A in iris sepal length. this is all false. If I sum this, this is 0, okay? I'm doing this operation to all columns of my data. So I'm doing this. I have no NAs in my data. And, okay, and let me check if we have zeros. It is. So we do not have any zeros, no zeros, no NAs. We apparently no outliers. We can, well, we'll see outliers, but later. I'm saving here an object called vars, and I'm removing the last column here because I do not want the species names. And I want to apply, I want to check the descriptive statistics of this data. So I'm applying the mean function to all columns. I'm applying the median function to all columns. This is the mode, the most frequent value. What I'm doing here is that I'm tabulating all this data. So this is the same table now applied to a numerical variable. I will sort this from the most frequent to the least frequent. And I'm grabbing only, I'm running this, I'm grabbing only the first one. So the most frequent value is 5, and I have 5 10 times. This is the most frequent value, the mode. Measures of dispersion, we can calculate the variance. We can calculate the standard deviation. I'm applying to my object variables, to all columns of my object values, the function SD. The SD is basically the square root of the variance. If I want to check if that is true, I can use again the function inside my apply. So I'm creating a function that is basically the standard deviation calculated by hand by calculating the variance and the square root of the variance. And if I check them, this is all true because this is the same thing basically. I'm defining here, I want to see the value of the coefficient of variation. Coefficient of variation is the standard deviation divided by the mean multiplied by 100. So I do not have a function for the coefficient of variation in base R. I'm creating that function. I'm calling that function CV. So this is a function of X. This could be anything. This could be vector. This could be any name that I want. But I'm applying a function to X, and I'm calculating the coefficient of variation, and I'm returning that value. If I apply this function to all the columns of my table, I have those values. I can check the quantile, so I'm applying the quantile to my entire table. See that the output here is different because we have the values for all the 4 quantiles, the default quartiles, and we can see the first, the second, the third, and all the quantiles here. We can change, there's an argument in the quantile function, so we can change the probabilities, the proportions here. So I'm in the 0%, 25%, 50%, my median, 70%, 100%. I can change that, and here I'm changing to any other value, 0.5, my median, and my 0.95, and this would change the output. I can check the range. So the range is the minimum and the maximum value for each variable. And another metric that people use is the interval. So it's basically the difference between the maximum and minimum value from the range. So if I grab my first like vars sepal length, I want the minimum, I want the maximum, vars sample length. This is the same that we get from range. If I want to see the difference between them, I could use the function diff. This will do 7.9 minus 4.3. This will be 3.6. So if I want to create my range function, I could create, so I will not create a function named range because it already exists. I will create a different function, my range, that will calculate the interval for each variable. The interquantile range is another measure of the range. This is the box of our box plot. We can calculate the correlation between the variables. So as we saw in the slides with the environmental variables, and we can use graphical methods. So this can happen because of my window. Let me do that again, probably now. Okay, so I'm plotting here how many observations I have for each species, Setosa, versicolor, and virginica. I can plot histograms with those. Oh, this is a bummer. My screen is too slow. Let me remove this. Shit. Okay. I will skip. Okay, I will show you each one of them. So I have the sepal length. I can see the histogram, sepal width. This looks much more like a normal distribution. Petal length. Oh, I want to do petal width here.
How do you put the organization? Because this is my result. Like this.
Yes, you should have that. I'm not having that because my window is too small.
But how you organize them? Like
okay. How we do that?
Yes.
We do that with this first command here. We are setting this function sets different parameters for plotting. It has tons of arguments. It has like a huge documentation. This particular argument will specify how you want to divide your plot window, and the numbers stand for the number of the rows and the number of the columns. Okay, so I'm building here a window with 2 rows and 2 columns, and then I'm plotting. In the end, I should always do this so I can set to the default 1 to 1. Otherwise, I will have like 4 windows for the rest of my workspace.
Yeah, if it depends of the order, yeah, because if you put first like petal length, or it is gonna be like, I mean, how you say I want this in the upper left, I want this in the right side.
Okay. There are ways to control that. I will show that in the graphics class, but the default here is what you are seeing, probably by row, right? But you can control that as well, okay? Okay, I will skip the histograms. Here is something good, a good thing to do. Here I have the density plot for the sepal width here. This is the density plot and the histogram. I can add lines to my histogram. So I'm adding this curve to my histogram, and I can check if my data is close to a normal distribution. Here, like box plots from these variables, we can do a box plot either by like plotting one variable, or I can use this notation to plot the same variable and create one box plot for each species. That is what we are doing here. Okay, I will go to this line 1, 2, 4, and we can follow this together because this is an important part of the class. So I'm plotting here the sepal width, okay? We can see, as I showed you, we have the median value, the first quantile, the third quantile. This side here is the interquantile range. So those are the extreme values that are 1.5 times the this value, this first or this third. And everything that we are seeing here, I can zoom in this here. Everything that follows out this interval is an outlier, and we can find that. So if we use plot equals false, I'm assigning this result to an object called my box plot. So my box plot here is, I have the statistics here. Those are the values that are being used to define those intervals. And I have here an object called out, an element. This is a list, and I have an element of the list with the outliers. So if I want to save my outliers, I can assign to an object called outliers the element out of this output when I'm using plot equals false. I can try and find where are those values in my table. So here I'm asking the values in the column sepal width that are in the outliers. This is a logical vector. If I ask which, this function which will tell me the position of those values in my table. And I can use those positions to extract the values that I want. So I can use this, and I can check where those outliers are. So I'm finding the outliers for all species together. I should probably do that for one species at a time. But they are like similar species. They should follow the same distribution. This could be an assumption here. And I could think of those values as outliers because they follow out of the range of the values that we defined. We can find them and we can see where they are. Okay. Okay. Let's follow what we have in it. No, I didn't remove the outliers. I could do that, okay, if I want. I do not need the which here. This would work without the which as well, because as we saw in other classes, we can use a logical vector to select parts of the object, or we can use the position here. I'm just showing you how we can do that with the function which, but it doesn't matter. If I want to remove those values, I could use the exclamation point here. So if I do that, I would have like all the values except for those who match the outliers values. And this could be my it is without outliers. I could save that to an object and I could work with that if I do want to remove those outliers. I'm doing a box plot now with the sepal width for all species together here. I'm doing this. I'm doing the same thing for boxplot 2. I'll probably want to find what this value is. I have another object. I'm doing the same thing, assigning to an object the value of the outlier. This is 2.3. This is what we are seeing here. And we know that the species here is setosa, so we could find what sample width values match the outlier value and only select the values from species setosa. So this is only one value that we are seeing here. And we can check if the data follow a normal distribution. I'm setting, okay. This is a function QUANTITATIve. This is the quantile plot for a particular variable. And here we want to check if this follows a normal distribution. So this is a Tukey plot, and this I can set a line. Let me do that for all of them. So this is always sample length for the 3 species here. I will zoom in because, oh no, this is terrible. This is also terrible. Let's look into this here. Okay, so we have here the theoretical quantiles and the sample quantiles. A Tukey plot will compare theoretical and sample quantiles. If we have a perfect normal distribution, all the values should fit this line here. So how much the data deviates from the line is a good way to check visually if the data follows a normal distribution. I have one line missing here in my Virginica. I didn't do this. Okay. So here, like we can see that setosa and Virginica are deviating a bit from the normal distribution. This is another way of doing this. We will do those kinds of plots to explore the residues of a linear model. So I want you to start using those graphics. This is the same as we saw in our in the slides, but we were exploring the environmental variables. Here, the different variables.
I see. In a different way to see if it follows a normal distribution, can we call some statistical tests for?
Yes, you can do that. There are specific tests for that. I like the visual first because you shouldn't do a test without visualizing the data. So sometimes you see the data and it's clearly not normal and you do not even need a test. But there are tests for doing that, and usually the idea is to basically compare how much in our histogram, like you have a particular variable, this is the histogram of the variable, and you feed, that's why I'm plotting this way, and you feed an empirical distribution. Each distribution has its own parameters, and you feed a distribution with the parameters from your observed values, and you can test how much these curves fits this histogram, basically. It's what those tests are doing. There's different ways of doing that. If you need a test, a p-value for that. And in the end, this is not something that we should do. I'm calling a package like in the basically last line of my script. This is not what we will do usually. This is exceptionally being done. And I'm calling the GG pairs function just because I like this function. It's super nice to see how the data are distributed. So here we have the density histogram, the correlation between two variables, and the value of the correlation for all the pairwise relationships. So there are a lot of other different explanations in the tutorial here. So you can follow the parts that I skipped. There's the explanation of the box plot as we had in the class. A little bit of more playing with the histograms, the beam widths of the histograms. You can explore that following like more slowly this script. But we won't finish the class without commit. So I have here, I modified something in my data manipulation script. I won't be adding this because I do not even know what I did here. But I save my script. I click here. I won't be using the window. I will do the old style way. Oh my God. So I will git add my R for EDA. If I check my git status here, I have a new file. I have a modified file here that I won't be adding now. I will add only that one. So I add, I will make my commit. If I do this, let me do this. And if I do this, oh, it's not working. But git commit my message. So adding script for EDA after class whatever. This is not a really good message here, but any suggestions? I'm okay. I didn't add. Git add RDA. Git commit message. Okay. And git push. So I can push my changes. And if I refresh here, I only have my other modifications. I did my commit. You should all do that. I will check your repositories later today, see how many scripts you have there. We all should have all the 4 scripts that we did together. And that's it. So tomorrow we'll talk about graphics. We'll be using ggplot, and I will show a little bit of RBase, but ggplot is a super powerful tool, and there's like no reason of doing things by hand on base if we have ggplot. And tomorrow I'll be here again from 4 to 5 if you need something. Today I won't be here. I will be here, but I can't be available. But tomorrow I will be here. And so see you tomorrow for graphics. Another class, another commit, and this is how we go. Thank you for today, and see you tomorrow.